On this trip through the digestive system, we will study secretion, the production and release of juices and hormones by the GI tract and its accessory glands. During the gastric phase of control, peptides in the stomach and distension of the stomach wall elicit a response. Will nerves, a hormone, or both mediate the response? Which hormone mediates this response? Note that we use the term neural reflexes when the neural mechanism is not fully understood, or when both long and short reflexes are involved. Remember that long reflexes to the stomach involve the vagus nerve, and that gastrin stimulates parietal cells both directly and indirectly by causing release of histamine. Will gastric secretion increase or decrease as the stomach begins to work? When a meal enters the duodenum, the intestinal phase of control begins. Distension, fat, acid, peptide fragments, and hypertonicity elicit a response. Will the response be carried out by nerves or a hormone? Think carefully about what the intestinal hormones do and what the appropriate stimulus for release of each might be. Which two hormones mediate this response? Fat causes release of CCK, and CCK affects gastric secretion. HCl causes release of secretin, and secretin affects gastric secretion. Will gastric motility increase or decrease as chyme fills the duodenum? Think about the function of the small intestine. This is the enterogastric reflex we first studied in the topic motility. The reflex dampens both gastric secretion and motility until chyme is neutralized and rendered isosmotic. Only then can maximal digestion and absorption occur. Excessive acid secretion in the stomach exerts negative feedback on gastric secretion. Will secretion increase or decrease when the pH of the stomach falls below 3? In addition to the regulation of digestion that is governed by food and the thought of food, the autonomic nervous system can exert control over the operation. What will happen to digestive activity when the sympathetic nerves are activated? What will happen to digestive activity when the parasympathetic nerves are activated? Parasympathetic activation increases digestive activities. Remember that the simple functions of the parasympathetic nervous system are to rest and digest. Hydrochloric acid and pepsinogen are actively secreted when the stomach prepares for and contains a meal. Once the meal has started to enter the intestine, feedback adjusts gastric secretion to maximize intestinal digestion and absorption. Most digestion and absorption occurs in the small intestine. The essential secretions for digestion come from the pancreas and liver, not from the small intestine. We will study secretions of these accessory glands first, beginning with the pancreas. The exocrine pancreas secretes a juice with two components that are regulated separately: enzymes to digest all major foodstuffs and a bicarbonate solution. CCK stimulates secretion of pancreatic enzymes, and secretin stimulates secretion of bicarbonate solution. Click the pancreas to begin. Pancreatic juice is important for digestion and protection. Click Digestion to learn more. Pancreatic juice is produced in structures called Acinar and carried through a network of ducts to enter the duodenum through the hepatopancreatic sphincter. Click the Acinar cells. Acinar cells secrete digestive enzymes for all major foodstuffs. They include proteases to digest protein, pancreatic amylase to digest starch, and pancreatic lipase to digest fat. Click the duodenum. The major pancreatic proteases are trypsinogen, chymotrypsinogen, and procarboxypeptidase. Like pepsin, they are secreted in an inactive form. Click the intestinal cell. Enterokinase, an enzyme embedded in the intestinal cell membranes, activates trypsinogen. Drag trypsinogen to enterokinase. Trypsin activates more trypsinogen. This process is similar to activation of pepsinogen in the stomach, first by hydrochloric acid, then by pepsin. Trypsin also activates the other proteases. Click enterokinase to repeat the animation, or click the duodenum to continue. Carbohydrates and protein are partially digested prior to arriving in the small intestine. Fat is digested primarily by pancreatic lipase in the small intestine. Therefore, if the pancreas fails to secrete adequate amounts of digestive enzymes, the major disruption in digestion is the failure to digest fat. Click Protection to continue. Duct cells secrete a watery solution containing bicarbonate. Bicarbonate neutralizes acidic chyme in the duodenum. A neutral to slightly alkaline intestinal environment is essential for maximal activity of pancreatic enzymes. Remember that the pancreas is a mixed gland with both exocrine and endocrine secretions. The endocrine pancreas secretes hormones including insulin and glucagon that regulate metabolic activities of the absorptive and post-absorptive states respectively. The major digestive function of the liver is the production of bile. Bile is produced continuously. When the hepatopancreatic sphincter is closed, bile goes to the gallbladder where it is stored and concentrated. Bile has two components that are regulated separately: organic compounds that are essential to emulsify fat and a bicarbonate solution. Recycled bile salts in portal blood regulate secretion of bile, and secretin stimulates secretion of bicarbonate solution. Click the liver to begin. Bile is important for digestion and absorption of fat and protection. Click Digestion and Absorption of Fat to learn more. The organic portion of bile is a mixture that includes bile salts, lecithin, cholesterol, and bilirubin. Bile does not digest fat. Both bile salts, which are derived from cholesterol, and lecithin emulsify fat. They keep fat droplets from coalescing, thus promoting enzymatic breakdown and absorption of fat. We will study digestion and absorption of fat in detail in the next topic. Cholesterol and bilirubin, a breakdown product of heme, are eliminated. Click the gallbladder to observe circulation of bile salts. Bile salts are recycled. Bile enters the duodenum during a meal and travels through the small intestine. Bile salts are then actively reabsorbed in the terminal ileum and returned to the liver in the hepatic portal blood. In the liver, bile salts stimulate secretion of bile. This pathway is called the enterohepatic circulation. Bile salts may circulate through this pathway several times during digestion of a meal. Click Protection to continue. Cells that line the bile ducts secrete a watery solution containing bicarbonate. This solution is identical to pancreatic bicarbonate solution and neutralizes acidic chyme in the duodenum. Secretin promotes secretion of both.